Okay, let us now discuss about the pre-fertilization events in androecium, right? And in order to do that, we have to discuss about the structure of the androecium. And in androecium, I have told you in the previous video that there are stamens, right? And stamens, it is made up of anther and filament. Now, if you look closely at anther, you can clearly see that anther is bilobed. It contains two lobes. Right? And each lobe is dithecous, which means each lobe it contains two theca. Theca it means covering chilone okay, so each lobe is dithecous. And anther contains 4 microsporangia, right? Anther, it contains 4 microsporangia. This is very important. You have to remember this, right? Anther nalola khazure. Anther nalola lob ni doa. Lob rare nalola tika ni doa. So anther nalola. The total anther khora nalo todo ganza nalola microsporangia khazure la wimbana. There are 4 microsporangia, which means there are 2 microsporangia in each lobe. So thika ki khar cover cheshayre microsporangium cover cheshayre. Each theca covers 1 microsporangium. That means since there are 2 theca in each lobe, it means there are 2 microsporangia in each lobe, and total there are 4 microsporangia. Now remember this microsporangia, it develops into pollen sacs. Pollen sacs lana pollen saksa gajiki ta regu doj lagora, sasa doj lagora. So microsporangia, they will develop into pollen sacs, and pollen sacs, they contain pollen grains. And ultimately pollen grains, they are male gametophytes. It contains the male gametes. Pollen grain nanglola male gametes diorta. So ये structure din nanglola ta wi imbai na anther di lobni gudupyo che lobni thongdo todo ni do ani din nanglola thika khajudo thika shithamba side rate ni do thika ani thika ki kimala khariore microsporangia yor. So total microsporangia shi to microsporangia chikni sum shiche ani microsporangia de shuma khar cha durelare pollen sac cha dur ani pollen sac na lo khare yore pollen grains yore and pollen grains, they are the male gametophyte. They contain the male gametes. I hope you are clear regarding the structure of the anther, right? So kisi kiranzugi lokte na lo remudina siwi by na, you can clearly see if you cut the anther de dusma tu by na, you will see that anther is bilobed, right? And each lobe contains 2 theca, and in theca it contains microsporangia. Ra ti kima jangush mi de ti khare microsporangia da microsporangia chik ni sum shilan do. Now if you zoom in, right, microsporangia chik ya chimbu sun ta wi by na, you will see that microsporangia, it is covered by 4 layers, right? 4 layers of cells. 4 layers ti khare khare rela wi by epidermis, endothecium, middle layers, and nanglo la khare yore tapetum la diyor. Microsporangia la di kima le yo microsporangia la di khari layer khazu gu tum sharan layer shigi epidermis, endothecium, middle layers, and tapetum. Now you have to know the structure and the function of these layers. Epidermis, endothecium, middle layer layers to the layer sum di lengyan kharelana to protect the microsporangium and help in the dehiscence of the microsporangium. When it becomes pollen sac, the pollen sac, they need to dehiscence. Dehiscence lana kam ni khache or pa khache or khache na ani pollen grain chine ma chilo tyon ru. So di chel or ko switch or lana layer dike sum gi chor. Nanglo la pollen grain gangsa develop cha ma sa phadola protection che. Pollen grain sama develop yabu cha sa veje la khache du zamalo khonzu sum gi ro chor. Epidermis, endothecium, middle layers ki. Whereas tapetum lash to tapetum lagi layer di these cells, right? These cells of the tapetum, they provide nutrition to the sporogenous cells or microspore mother cells, right? Microspore mother cell develop cheve kabla nutrient to suit or la vimena the tapetal cells ki da. So the example yang se ligduta, you have to know the function of tapetum, right? Tapetum la cell dige nanglo la khare, they contain nutrition and it provides nutrition to the developing microspore mother cells. So this diagram is very important. You have to study this particular diagram. It is the structure of microsporangium. Okay, DJ. The microsporangium nanglo di nanglo yogi sporogenous tissue lash to sporogenous tissue nanglo kharto all these cells of the sporogenous tissue nani kima dro. So all these cells of the sporogenous tissue, they are microspore mother cell, which means they have the potential to become microspores or future pollen grains. Pollen grains chaya gi nubayor. And pollen grains chaya gi nubayor yo de la nuju su de or la wim na tapetal cells ki te or. And later on, they all become pollen grains, and later on what will happen, the pollen grains will burst out. Pollen grains the hachire din ma thendur di ma khache gi process to di langa khala gre dehiscence lagta. Okay, so pollen grains are released through dehiscence of the pollen sac. And pollen sac dehiscence chala su robachi korla so epidermis, endothecium and middle layer cell digegi robachi gor. Clear chung so you need to understand that these diagrams. Now there is one process during the pre-fertilization event which occurs in the androecium. Androecium now pre-fertilization event microsporogenesis laadi. It is like a gametogenesis, gamete formation do chador, a male gamete formation do chador. So it is called as microsporogenesis. So microsporogenesis ki definition dikhare la the process of formation of microspores from the pollen mother cell through meiosis. It is called as microsporogenesis. Di nalo yo cell jo sporogenous tissue nalo yo cell dige samanya kharala so microspore mother cells rela so. So they all have the potential to become microspores, right? These cells, they will undergo meiosis, right? Undergo meiotic cell division, and they will give rise to microspores. So microspores dige shuma kharcha dure. Microspores, they will develop into pollen grains. So if you look at a single cell, this cell jiglata, this cell the meiotic cell division choi baina meiosis. If this cell undergoes meiosis, it will give rise to 4 cells which are haploid in nature. Quote diploid in baina meiosis ki jelang azul khar ragre 4 daughter cells ragre. These 4 daughter cells, they are haploid and they are called as microspores. And these microspores, they will further develop into pollen grains. I hope it is clear, right? Todan the seu chung so microspore mother cells ma upe to dige sama kharchi re, they will undergo meiosis and give rise to microspores, and microspores, they will develop into pollen grains. Now if you look at the pollen grains, I have told you that they are male gametophytes. Pollen grains, they come in various sizes and shapes. Okay, tishi manda kajigi pollen grain gi sopta ta ro chiju manda yor. Okay, so their morphology differs from species to species. So you have these diagrams shown in your textbook. Diagram all these are pollen grains of different plant species, right? The common pollen grains, they contain 2-layered wall, right? Pollen grains wall layering layering exine and intine. Very important. From examination point of view, this particular point is very important. Pollen grain the cover jomarwe. Pollen grain ki chilo ki chishin chorwa. The 2-layered race. Layering the exine and intine line. Ta nedon kechambul di exine la di exine lana chilo de la. The outermost covering of the pollen grain is called as exine. And exine, it is very, very tough. Exine, the bit of your. It is very tough and call a toughness the karigite or what renders exine so tough is it is made up of sporopollenin. Exine like the karig dupsha yore, sporopollenin like the substance chik dupsha yore. And sporopollenin like the yoit of your kachin tough yore it is extremely resistant to adverse environmental conditions. Right, and there are no enzymes which is known to digest sporopollenin. Sporopollenin digest chetungi ki enzyme thada bod ha ko homar. Di imba sonza pollen grain the exine gi tum shayro. And ko yo thabu chador, it becomes really tough, right? So inside the male gametes can survive for a very long time. But at certain regions, certain regions now there are pores known as germ pore, where the sporopollenin is absent. Sporopollenin yosa che ani mesa saja marwe exine gi yelang azu khat lagre germ pore lagre, right? And germ pore, they are necessary for the pollen tube formation. Pollen tube form chid example jump or yogor. Isi chazan spore pollen in gitum shawe imba na male gamete ma thuntu marwa. Male gamete ma thunsa ki sacha dikha lagre jump or lagarta. So you have to remember that. Okay. So dinanglo la kharre to exine dikhari ki soyor lasun sporopollenin. You have to remember this word as well. And sporopollenin is very resistant to external adverse conditions. Acid inere, base inere, khari imba inere, khola ne ke tu bomar. And even there are no enzymes which are known to digest sporopollenin. And inner thin layer the inner thin layer is called as the intine. Right? Intine is very thin and it is a continuous layer, unlike exine. Exine continuous layer marwa. There are germ pores. Whereas intine is a continuous layer and it is made up of cellulose and pectin. Right? Intine lady cellulose the pectin ki suesha yore taptaj mada yomar. Exine lady tupu and it is made up of sporopollenin. I hope you remember these points about the pollen grains ki structure kala. Now, pollen grains, when it becomes mature, microspore ne ko khali khar la re, pollen grain la ra. When the pollen grain matures, it has got two cells inside. Mature pollen grains, they have got two cells inside, and those two cells are vegetative cell and generative cell. Pollen grain rare na cell ni yorta, cell ni kharre la vegetative cell, the generative cell. So this is a mature pollen grain. So this is a mature pollen grain over here. Inside the mature pollen grain, you can see vegetative cell which is larger and a smaller generative cell. Vegetative cell has got abundant food reserve. It has got larger nucleus. Whereas generative cell, they are smaller, right? And they float inside the vegetative cell. Generative cell dikhachi to chunga to ani vegetative cell ki cytoplasm nanglo la kill de yoros de do. Now, over 60% of the flowering plants, they release their pollen in this two-cell stage. Angiosperm kya thukjul hazam ji khaji gore konjo ki pollen grain di two-cell stage nal malyo sha gore. Whereas another 40% of the angiosperms, their pollen grains contains three cells. Three cells khande chagre la wimba na. The generative cell, it divides into two male gametes. Right before it lands on the stigma, the pollen grains are released in three cell stage, in which the generative cells will divide into two male gametes. Clear chungswa? In 60% of the angiosperms, the pollen grain is released in two cell stage, whereas 40% of the angiosperms, they release their pollen grain in three cell stage, in which the generative cell, it divides into two male gametes. Okay, so DJ. Further miscellaneous informations regarding pollen grains. Pollen grains, they can cause severe allergies to some people, leading to respiratory diseases such as asthma, bronchitis, etc. Right. For example, example pe nenga chumburda Parthenium liger, common name khare carrot grass. Loptanal dendu ke do ngazu ki TB method lagdo. Di khare di Parthenium layer. It is an invasive species. Invasive species chichadu. So medo di gi pollen grain larwa. Pollen grain gi mi mangbuchi la khachi allergy cause chiwor. Asma naya da denso. So pollen grains of this particular plant can cause severe allergies, leading to respiratory diseases such as asthma and bronchitis. Pollen grain pollen grain ki some of the pollen grains, they can be used as food supplements because pollen grain, they contain nutrition. Pollen grain nutrition pollen grains, they can be used as food supplements, and they are claimed to enhance the performance of athletes and race horses. Race ani athletics sports person ki performance di yarge ani pollen ki food supplement muscle build up ani protein powder athletics athletes ki pollen food supplements sawa na ani ki performance claim they are claimed to enhance the performance of athletes and even race horses. The for the crop breeding programs, some of the good pollen pollen in pollen banks. And this pollens can be stored in liquid nitrogen in minus 196 degrees centigrades. Pollen grain we can store pollens in the pollen banks, and these pollens can be used for crop breeding programs. Crop breeding pollen it's just like male gametes. Male gametes sperms store plants pollen grains store pollen banks and later on uh, they can be used for breeding programs. So that is some miscellaneous information regarding the pollen grains. So can also clear pollen grain for the so pre-fertilization events in androecium, it involves microsporogenesis, right? And microsporogenesis leads to the formation of pollen grains, and inside the pollen grains we have got male gametes of the plant. In the next video, we will discuss about pre-fertilization events in gynoecium, or the female reproductive part. Okay, thank you.